Okay, Lamai, we learn up to here in the 20th lesson. Then this Punnett square, how to draw the Punnett square. Now let's go move forward. Select another pair of contrasting feature of pea plant using 20.1 table. From this table, they are asking to select another pair of feature character as an example color of the flower and do this punnet, do this drawing. State pure breeding dominant character, so you have to identify the pure breeding dominant character, then pure breeding recessive character using symbol. Okay, you have to draw the pure breeding dominant character. Okay, write down, and the pure breeding recessive character using symbols. Build up a Punnett square, then the Punnett square to show what? The inheritance of this monohybrid character. Explanation of patterns of inheritance using probability. Explanation of patterns of inheritance using probability. When any two different objects combine randomly, the result will take place according to a particular pattern. This is the pattern which I draw with you here. Here, see, this is capital T, simple T, capital T, simple T. So from this, you are going to write this first two at first, then first one in the first pair and second one in the second pair, then the second one in the first pair and the first one in the first pair and the second one in the second pair and the second one in the second pair. You see how it goes like this. These are the combinations which could be occurring here. So the same thing happens here. As an example, consider random combination of x and y objects. Okay, random combination. How we draw it? This x goes with this x and also with this y. So xx plus xy, and this y goes with this x and also with y. So xy, yy. Two characters present in parent inheritance. Sorry, two characters present in parents inherit into offsprings randomly. Carry out below activity to identify the probability. Probability means the ability of an incident to happen. Do you know the probability of getting the head or the tail when tossing a coin is half? Is that true? So think if you are tossing a coin, uh, it could be get its head or the tail. Head or the tail. So the probability is 1 over 2 because there are two options and you are getting 1. So it's 1 over 2. Got it? And the bed experiment is similar to relationships of probabilities using two coins at a time. To find out head to head, head tail, tail head, tail tail probabilities, the probabilities obtained from each coin has to be multiplied. Then the probability is half into half. So it's 1 over 4. When the same incident takes place in two ways, getting head, tail and tail head, the total of separately taken probabilities is 1 over 4 plus 1 over 4. So it's 1 over 2. Anyway, it has two options. So we have to go like this. Identification of probability patterns obtained by bed experiment. Divide the class into 4 to 5 groups. Provide 2 vases with mixtures of 50 white beds and 50 red beads to each group. Instead of beads, you can use buttons or seeds with 2 different colors can be used. So this is an experiment. So from here you see they are doing an experiment to find out what has done by Mendel. Here the class was divided into 4 to 5 groups. The first group, then provide 2 vessels with mixtures of 50 white and 50 red. So the same number of 50 white, 50 red. Okay. Then take out a bead from 2 vessels at a time and note the color of 2 beads with tally marks in the relevant column and return them into the same vessel. You have to take 2 a bead from 2 vessels. So for each group they are giving 2 vessels and each group, okay, 2 vessels and 50 white and 50 red. So we identify 50 white as W and red as R. So 2 vessels. So you are getting 1 from each. So both 2 vessels contain 50-50 white and red. Okay. So you are in a one group. Then what you get? You are getting 2 vessels. Each one contain 50 R and 50 W. 50 white and 50 red. Okay. So you are getting 1 from each vessel. Then what happened? Then if you get at the first time, if you get both are in red, you have to mark it here. Both, then one is red, one is white, put a mark here. Then one is white, one is R, put a mark here. The first one, if the first one is white, you have to mark here. And the second one is red. If the first one is red and the second one is white, you have to mark here. And if the both are white, you have to mark here. Okay. Fill the above table using results obtained by the above practical using tally marks. Calculates times of red beds and probability of getting red bed. Times of red white and probability. Times of white red and probability. Times of white and probability. Using those probability values, find out the ratio of RR, RW, WR to WW. Let's analyze the Mendel's results using the probability results obtained by this experiment. Okay, the methanol we did that experiment of using this red and white beads. But when it comes to Mendel's experiment, he used tall plants and short plants. So that's what he has done. So the probability of getting red, it ended up as 1/4, white 1/4, red 1/4, white red 1/4. So see this white red and red white both are 1/4. So it means red white. So here red again 1/4, white 1/4, red white or white red, we can consider them as getting same. So it is 1/4 plus 1/4. It means if you add this 1 in the 1 above the 4, it becomes 2 and divide 4. So it ended up as half. So if you write it like this, you can cut all the 4s and find out the ratio is 1 to 2 to 1. 1 to 2 to 1. So according to the Mendel's monohybrid cause, the ratio of F2 genotypes, F2 is like this. Similarly, the probability ratio of bead experiments compatible with the genotype if you did that experiment in real locations, you get this 1 2 1. So the first one is both capital T, the last one is both simple T, and the middle one got the TT got the 2. Basic concepts of genetics. Okay, we've done with that. So what they ask you is, for F2 generation, what is the ratio? So 1 to 2 to 1, 1 to 1, and their genotypes are T, capital T, capital T, simple T, and simple T, double simple T. Now we come to the basic concepts of genetics. Gene concepts about inheritance. The things which we have learned, okay? Gene concepts. Mendel said that features of an organism is determined by special particular factors. The identified particular factors is named as genes later. So they are the ones we are calling as genes. Features of an organ determined by factors. These genes are the factors which determine the determine the feature of an organism. Okay. So here determined by. Features determined by genes. Okay. In gene concept, as a standard, the dominant feature is denoted by a capital letter. If it is a dominant, highlight or underline these things. It should be denoted in capital letter. Okay. And if it is the recessive feature, it is denoted by simple letter. Okay. We have already used these letters in Mendel monohybrid cross. Yeah, we did it, Punnett square and monohybrid cross. If two genes are similar for a given feature of an organism, it is said that this organism is homozygous. Yeah, we discussed these things already. Here they are mentioning it again. Else the organism possesses or else that organism possesses homozygous genes. If two genes are not similar, then that organism is heterozygous, or else organism possesses heterozygous genes. See, example, gene for the round seed is capital R. That means this is a dominant. Okay, so this should be the dominant. Gene for wrinkled seed, it is simple R. So this should be the recessive. Okay, I think you got it. This should be the recessive. Then homozygous situations, it's both should be same. No? So it could be both, could be dominant genes, and in the other way around, both could be the recessive gene. So it is RR. Anyway, they are homozygous, no? The same type. Homozygous. And simple double R. And what about these two types of genes? They call as heterogeneous, and it can be written as capital R, simple R. When it comes to tall and short, it's capital T, simple T. Gene expression, the combination of a gene pair for a particular character is known as gene expression. The combination of gene pair for a particular character. Okay, what is that? Particular character. Think like the character is tall. Character is tall. Okay. So what is the gene? What could be the gene pair to express that tall character? For tall, it could be dominant, both dominant. That means homozygous. That means capital T, or capital T, simple T. Both could be go with that character, no? So gene expression can be capital T, simple T, or both capital T. That means double capital T. If the character is short, the particular character is short, what is the gene expression? It should be double simple T. Got my point? That is what we call as gene expression. Then phenotype and genotype. The feature that externally appears is known as phenotype. That means I told you that if it is a towel, that is a phenotype. And genotype means a gene composition. To determine that feature is known as genotype. The gene composition. Gene composition එක තමයි genotype කියන්නේ. හරිද? As an example, they have given. So the feature that externally appears is the phenotype. So they have given it below. See, the phenotype of a round seed heterozygote is round. The phenotype of a round seed heterozygote. It's a heterozygote, okay? Round seed. What is round is dominant or? See here. Round seed. Shape of the seed. Round wrinkle. Round. So 3:1 ratio. Round round is the dominant. So round is the dominant. Okay. So wrinkle is the recessive. So you have to write capital R because round is the dominant. So what they are asking here, what they mention here, the phenotype of round seed. So round seed, capital R is the dominant one. Heterozygote. So heterozygote means if it's homozygote, it's both capital R. Due to this word heterozygote, you should write the phenotype should be the round shape, but the genotype is capital R, simple R. Why? Because of this heterozygote characteristic. And round seed homozygote is round. So here the genotype is capital R because it's homozygote. And when it comes to the phenotype of wrinkled seed homozygote, because wrinkled seed cannot be in heterozygote. When it comes to heterozygote, it could be the round seed because capital R, that round shape is the dominant one. So if dominant gene and both dominant and recessive seeds are present, the phenotype should be that dominant characteristic. So it is RR. If you have questions, please ask me after you hear to this one. Nature of genetic materials and genes. We have this one then. Okay, I'm going to teach you it in the next part. So I'm going to stop here, this part. Okay, thank you.